Good morning and welcome to the 2011 Royal Tyrrell Museum Speaker Series. Today, the Royal Tyrrell Museum and its cooperating society are proud to present Dr. Takuya Konishi. Takuya is a postdoctoral fellow here at the Royal Tyrrell Museum. Takuya began his undergraduate studies at Kochi University in Japan, but after his first year, he transferred to the University of Alberta, where he obtained a bachelor's degree or bachelor's degrees in earth sciences and biological sciences. He stayed at the University of Alberta to pursue his PhD in biological sciences, where he studied the systematics of Platecarpine mosasaurs. In September of 2009, Takuya joined the Royal Tyrrell Museum as a postdoctoral fellow. Takuya's research interests revolve around the evolution of mosasaurs, a group of extinct marine lizards that reached a worldwide distribution during the Cretaceous. As such, Takuya's research has taken him to museums all over the world, including those in Canada, the US, France, Belgium, England, the Netherlands, and Japan. Today, Takuya will discuss his involvement in the study of an exceptionally well-preserved mosasaur skeleton in a talk entitled "The Tale of a Mosasaur, a Window on the Tempo and Mode of Evolution in Aquatic Tetrapods." So without further delay, I present to you Dr. Takuya Konishi.
Well, thank you very much, Michael, for your wonderful and always very polite introduction and kind words. This morning, I'm very pleased and happy, in fact, to be able to share with you what I would consider one of the most exciting research projects that I was involved in. And this constitutes this particular organism called Platecarpus, and that's one genus, one group of mosasaurs, which we know from about 60 species today. Mosasaurs, of course, lived in the water, as you can see in this fantastic reconstruction by Professor Takashi Oda in Japan, who works at the University for Fine Arts. But I tentatively hid the end of the tail. I will talk about the tail of the tail towards the end of my talk. Okay, so the tail of a mosasaur, a window on the tempo and mode of evolution in aquatic tetrapods. There are certain jargons such as tetrapods, but I would like to clarify that as we go along. So first, I talked about tempo and mode in evolution. So what's evolution? From the very, very long time ago, it's simply put, it's descent with modification. We are talking about organisms or animals and plants and so on. They change as the time progresses and becomes new species and so on. That particular wording, descent with modification, was in fact used in Darwin's The Origin of Species. In fact, Darwin seldom used the word evolution. In fact, I don't think that he did in his Origin. He always used modification as the term in lieu of evolution. But we can certainly synonymize them today. There are two main categories of evolution in biology, microevolution and macroevolution. I think I'd like to talk a little bit about those two in the next slide. But before that, do you know what tomorrow might be? It's February 12th. What's that? Right. It's Darwin's 202nd birthday anniversary. And so, happy birthday, Mr. Charles Darwin. Okay. Okay, so microevolution is basically we are only concerned about a population, not even a species of organisms, and talking about change in gene frequencies over generations in a given population. For example, here is a group of hypothetical insects with 3 different colors. Here, if we assume this is a population we have today and now, we have two more individuals of, which color was it? This yellowish color bugs, two more than other colors. Now, depending on the situation in the environment of the time, this color composition may change. Not to say that they are not the different species; they are all the same species, but the expression, for instance, the color change and fluctuates through time. Sometimes browns are more dominant than other colors. Sometimes the beige color is more dominant than other colors. That's what they are talking about, microevolution. And microevolution is really the fundamental phenomenon that would lead to speciation, a creation of a new species, derivation of the new species. That's microevolution, but in paleontology, it's more often, and in fact quite suited to talk about macroevolution. Macroevolution, we are talking about much greater scale. So it's evolution of organisms over a geologically long period of time. And we are talking about the phenomena such as a evolution of flowering plants, evolution of first terrestrial vertebrates, evolution of flight in birds, and so on. So that naturally involve more inclusive taxonomic units. We are not talking about species, we are not talking about certain genera, but we are talking about families, orders, and so on. A class, mammalia, for example. So that's what it means by above genera. Here is one example, a fossil exemplifying the influence that could be made on macroevolutionary thoughts for us, because this one is currently the most tetrapod-like fish. Tetrapod, again, I had that word in my first slide. The tetrapods are basically vertebrates that came onto land, including ourselves, and amphibians, salamanders, and reptiles, and alike. So Tiktaalik is actually it's from Nunavut, Canada, and it's unique amongst fish because it has a freed pectoral, girdles that is free from articulating with the skull. In fish, all the fish, you always have pectoral girdle, your shoulder girdle, connected to the skull, so you don't have the neck that can move. But here, it's very special in terms of not having the neck, in other words, mobile skull, thanks to the separation of the head from the pectoral girdle and shoulders. So the nickname is fisherpod. We'll come back to Tiktaalik later on. And then in this talk, I would also talk about a tempo and mode of evolution. Now, back in 1944, George Simpson, who is renowned paleomammologist and evolutionary biologist, had this book out. And the very first sentence of this book starts as, how fast, as a matter of fact, do animals evolve in nature? And he admits this is such a great question, but also very difficult one. And his definition for rates of evolution was amount of morphological change relative to a standard. This standard can be many things, but for example, we could have an absolute time as a standard so that you can compare the morphological changes through, say, 1 million years of Earth history in various organisms. And we are also talking about morphological change because in reality, we really can see morphological changes in fossil record. So we are not concerned with genetic changes in this case. But that's okay, they are related to each other. So just because I talked about absolute age, I think I thought I was going to show this diagram here, and with many arrows, but you can ignore them now. Basically, this is the Earth history, and nicely divided into those 4 eons. And Paleozoic is where the Tiktaalik comes in. So that's the, and also the first reptiles, such as Hylonomus, comes in. And then these yellow lines indicate the terrestrial or somewhat terrestrial land-living organisms. So Tiktaalik was the one around 375 million years ago that ventured onto land or becoming a tetrapod from fish. And only in a matter of a million years, 100 million years, or less than that actually, 50 million years or so, you have first reptile. So many tetrapods diversified in this period of time on land. However, as early as 290 million years ago, this strange reptile called Mesosaurs, it's not Mosasaurs, Mesosaurs. All the students get tricked by that question from experience. But Mesosaurs, it went back to water. Now, this ancestor didn't used to live in the water. It used to live on land. It's only that Mesosaurs came back to water secondarily. Now, that trend continued on at the early Triassic, in the early part of Mesozoic. These sauropterygians went back to the water, so were ichthyosaurs, such as Utahtiosaurus, shown here. And then the list goes on. Here's the marine crocodile that went back to water, in the early Jurassic. And then this is the relative of Tuatara in New Zealand. Plesiosaurs is the aquatic group of sphinodonted reptiles. So that again, all the ancestors of these groups were living on land, went back to the oceans secondary aquatic adaptation. And then turtles, they were not the last one to when going back to the sea. Around 110 million years ago, you can find the first sea turtle remains in the fossil record. And then, boom, finally, mosasaurs. You can see at this point that mosasaurs really were one of the last lineages of reptiles during the Mesozoic, the age of reptiles, that went back to the sea. And of course, all of those marine reptiles are not related to each other. At all. They were independent events throughout this long period of time during the Mesozoic. At the end of the Mesozoic, all of them except for marine sea turtles went extinct. And of course, the age of reptiles finishes. And then in the Cenozoic, now the ocean is vacant. Some mammal group thought, hey, that's a good place to go hunt for food. So the whales came in. So again, all of those things were really sporting what Tiktaalik did many years ago. And because Tiktaalik was just trying to be land-dwelling animals, trying to make the limbs out of the fin and so on. But then it did quite the opposite in the different timings in the Earth history after that. These red arrows just indicate where mosasaurs, how long mosasaurs lived. So from its strictly late Cretaceous, from about 98 million years ago and 65 million years ago. This slide, I showed it last year as well, but as I just said, there are so many reptiles in the sea during the Mesozoic. So what we see today, we are all familiar with those, but predominantly on land, could be seen in the water during the Mesozoic. And here, not only those groups in the Mesozoic, we have two main groups of marine reptiles that were strictly Mesozoic, and we have no living relatives whatsoever of both plesiosaurs and ichthyosaurs. So in fact, Mesozoic was full of marine reptiles, and that was very different from what we see in the marine realm today. Really different. So, without further delay, I'm going into the introduction to mosasaurs, Mosasaurs 101. Okay, so they were a group of squamates, lizards and snakes, that is, that lived in the world's seas, oceans, and even bodies of fresh water about from 98 to 65 million years ago. Here at the end, at the bottom of this column, I'm showing what mosasaur ancestor might have looked like. This is not exactly a mosasaur. It's called a Aigialosaur, and Aigialosaur is what we call a sister taxon to mosasaurs. But the skull morphology, the head of Aigialosaur, really is very similar to what we see in these gigantic mosasaurs. Now, this one is only about a little over 1 meter long in body. The tail is truncated, but it had a straight, long tail, tapering tail. And here we are talking about a mosasaur of anywhere from 3 meters to 10, 12 meters long. That's all big thing. So, and they are also the only squamates to have possessed flippers instead of or deriving from the limbs. So even in this Aigialosaur, while it was aquatic, it still has these short limbs. Here in mosasaurs, fully derived, you have flippers instead of limbs. That's really only good for steering yourself in the water so that you can swim up and down and left to right. Squamates, what are they? They are lizards and snakes, and we have lots of them. In fact, they are the most diverse group of reptiles we have today. 3,000 lizard species and 3,000 snake species are known, approximately. And now, in the fossil record, what we have is our bones. So let's see what the squamate skull looks like. Basically, this yellow part is empty. Where in other reptiles, this portion usually has a strap of bone that forms the lower boundary of the skull. This space is open in squamates. So that's a good, generally holds true. And because of that empty space, some people, well, some squamates do have this bone called quadrate, being able to move back and forth, having this red star portion as a hinge. So because there's nothing to stop this bone from sliding anteriorly, this does move anteriorly, and that is employed in some of the biting motion in squamates. And that's called streptostyly, but doesn't hold true for every squamate. We don't know if streptostyly was present in mosasaurs either. Now, if you're lucky and have a exceptional preservation, then you sometimes find a scale impression of mosasaurs in the fine sediment. This is 84. So 84 million years old Tylosaurus, big mosasaur scales, which I kind of aligned with today's reticulated python. And here what you can see, important feature is that the fact that those scales are imbricating. They are on top of each other. So, and just like fish scales, and that's what you see here. Now in this case, they even have more similarity where you have a diamond-shaped scale as here. And then even this median keel, those straight lines that occurs in the middle of the scale, you can see that here on the snake's scale. Not all the snakes show this. It just happens to be so. And moving along, I just briefly said earlier, but they were indeed the largest squamates ever lived in the Earth history, and some attained a body length of 12 meters long, and maybe even more. So the size matters. These are taken in 2005 2004 and 2005, and that's me as a scale, a little bit younger, but not much. And this is a skull of a Tyrannosaurus pruriger. This eye socket, lower jaw, teeth pointing to me. It's dead, thank goodness. And then Mosasaurus conodon, this is a skeleton. The tail is going backwards, but again, this is a big, big-headed lizard with flippers. Here is just a single, one side of the lower jaw of another Tyrannosaur individual, and this is my foot. Now, I believe I used to have much bigger boot, and this was a size 12 or so. So you can see how big that thing is. And the teeth were also really big. And they could eat many, many things as they wished. And then, speaking of the food, they ate fish of many sizes. They ate other marine reptiles, because there were many of them in the Mesozoic, including mosasaurs of smaller sizes and plesiosaurs, some of those Loch Ness Monster style plesiosaurs were consumed by not the Loch Ness Monster, that's mystical, but looks like a Loch Ness Monster, or Loch Ness Monsters looks like plesiosaurs. Either way, they were eaten, and then clams. Now, in addition, they never came back onto land to lay eggs. Maybe they were wondering if mosasaurs were reptiles, they should have laid their eggs on land, like sea turtles today. Now, we don't think they did so. Now, I'll explain that later. So food for mosasaurs. This is a hip region of a big tyrannosaur, about 8 meters long or so. And here are a jumble of bone, hashes of bone. And you can see all those little small round things. They belong to a smaller mosasaur that was consumed by this mosasaur. And this is the bone that I talked about a little bit earlier, quadrate, and of another mosasaur. This individual big mosasaur also had a 1.2 meter long fish and also diving bird of considerable size. So it was really hungry until it died, which is a good thing, I think. And then food for this is another mosasaur called globidens. And globidens means round tooth. Obviously, they had round teeth. What was it doing with them? Well, they were using those round teeth to crush clams. And so there were shellfish-eating mosasaurs, also rare in the history of Earth. I said that they didn't come onto land to lay eggs because here we are showing the reconstruction of gravid female Platecarpus, in fact, about more than 6 meters long, giving birth to about 1.5 meter long or so offspring in the ocean. This individual was found with as many as 6 small but same-sized, same species individuals, mosasaurs, inside. And each of the small individual was about 1.5 meters long in length. That is too large to be contained in an egg from any reptilian standards. And they were not the food item of this mosasaur either, because they were all the same size, and 6 of them is too many. So the natural way to think about it is that it gave live birth in the water, which is not uncommon in squamates, because many squamates do give live birth even on land. And in fact, Aigialosaurs that I talked earlier, the more ancestral form of mosasaurs, they are known to have given birth to live young as well, based strictly on the fossil evidence inside the abdomen. So that never holds, it does not hold true anymore. And then there are 60 species of mosasaurs known within 30 or so genera. And, but despite their high abundance in fossil record, shall we say, we don't still agree, most workers don't agree with each other in terms of where really they belong to in the group of squamates. but the general consensus is that they are angulomorphan lizard. Here's the a phylogeny of lizards and snakes here, so squamates, and then the sister taxon being Tuatara from New Zealand, and mosasaurs will be nested somewhere here in Angulomorpha, but not all people think that they are Branoidea. But it's safe to say at this point in time that mosasaurs were some kind of angulomorphans. We'll leave it there because today's focus is not the phylogeny, but evolution, functional aspect of mosasaurs and macroevolution of aquatic reptiles. So the locality of this fine specimen that I'm going to talk about for the rest of the talk is Kansas, western Kansas, which is just peeking up there. And it's no surprise to find mosasaurs in our continent because during the late Cretaceous, the Gulf of Mexico and Arctic Ocean was connected with this inland seaway called Western Interior Seaway. And they were there most of the time. They receded towards the very end of Cretaceous, but when mosasaurs were around, they were there for the most part. And in fact, here's the Alberta. Here's Alberta we have, and then half of it, Alberta gives to dinosaurs. You know, it's land. Dinosaurs did not live in the sea, but in fact, more than half of it was for mosasaurs and other marine reptiles. So I think to understand Alberta paleoecology during the Mesozoic or even late Cretaceous, it's crucial that we do understand both terrestrial and marine fauna because you can see Alberta was under the sea more than 50% during the late Cretaceous, more or less. This is a specimen, and I raised my voice because I got excited, in fact, because I still do. Why do I get excited? Well, it's because this was one of the last specimens that I looked at in my dissertation work. I made many trips to different museums and went to look at more than 400 individual specimens of this particular group called Platecarpus. And many of them only have a skull or even a part of the skull or a paddle alone. And even if you have the backbones with the skull, they are always disarticulated. And little had I imagined that the world's finest, not only Platecarpus, but mosasaur specimen was in the West Coast, near Disneyland. I don't know why. Away from Kansas or Midwestern North America, sitting in the Western coast. And one day we suddenly got a contact from the curator at the LACM, Los Angeles County Museum, Luis Chiappe, about their fine mosasaur and whether or not we are interested in studying the specimen. The answer was yes. So this is a fine specimen, not only because it has a beautifully articulated skeleton, but also the preservation is so exquisite that it shows many unexpected features at the final level. These top three pictures show photographic images that were taken under the UV light. To easily explain it, you know how people at the customs verify your passport if it's real one or not by putting it under the UV light, and suddenly this green mark comes up, and that's a real passport. It's a true one, and if it's not, it doesn't. What we are doing here is quite similar technique, where many features are not visible under the natural light and in your naked eyes, but some elements or minerals that replace the Platecarpus can give off the light, under the UV light. So although we couldn't see this structure here, which I'm going to explain later, these you can see, only you can see is kind of a vague pattern on this bone called frontal. But under the UV light, you can start seeing those interesting structures. B is showing what the scales looked like on the forehead of this mosasaur. This is the forehead, this is the eye. And you can see this is hexagonal and rather large. I believe this is 1 centimeter. Actually, this is 2 centimeters. So some scales on the head of Platecarpus attain about 1 centimeter. And that's big compared to body scales. Here, this is where the nostril was. But of course, not all the space was open when it was alive. The only portion that was breathing the air through was this little bit expanded portion where we detected this line, which we interpreted as the neural slit, if you will. And the slit is, I think, a good word because mosasaurs were living in the water, and they have to shut their nostrils when they're in the water so that they don't swallow the water from the nose. We know that very well. And then here is showing another, this part of the, just in front, right in front of the eye, we have a different shaped, a bit more rhomboidal shaped scales instead of hexagonal shaped scales. And the significance of that, we do not know for sure. I don't. Here is a series of shots. This is under the normal light, and you can still see it. But this is a windpipe. And windpipe, if you kind of feel your throat, you feel kind of a bumpy structure here. That's your windpipe. And it's bumpy so that it doesn't collapse so easily. You have these cartilaginous rings, row of rings that is protecting your windpipe so that it ensures the airflow all the time. And we see that very rarely in fossil record, but this specimen most certainly shows them here. Very, very, very good specimen in preservation. And then let's focus into the eye of the mosasaur. This is, again, a little bit of a playing shot with UV light. This is normal light. What you see here, inside this eye socket, you have another round structure here. That's a bone that protected the mosasaur's eye called sclerotic ring. And that ring does not exist in our eyes, but many reptiles, including birds, do have them, including dinosaurs. And when you don't have them in the fossil record, that's because they were gone by the preservation process. So this was well preserved enough that to retain the sclerotic ring quite completely. And here is the sclerotic aperture through which the mosasaur was watching outside its world. And zooming into this sclerotic aperture, we see this kind of purplish bumpy structure and looking very organic. And we see nowhere else this kind of bumpy structure nowhere else on this fossil, although it is well preserved. And upon examining this portion as one sample under the high magnification scanning electron microscope, we see this consistently aligned and also consistently shaped and sized globular structure. And that's like a few microns in size, and what we consider that is they are pigments of the lining retina inside the eye that was kind of came forward when the animal got crushed and pushed down against the sediment it was lying. So, and we see nowhere else this kind of structure in different parts of the mosasaur. So we don't think these are microbial cells that were harvesting on mosasaur's decaying structure, but rather this is a mosasaur's eye structure, the pigment structure. And I think we made a reasonable argument there. Moving on to the body. Body scales were more or less diamond-shaped rather than hexagonal. And they are also smaller. Next slide. So those are representative body scales. Some are 3D in preservation. Some have what seems to be a differential preservation of the parts of the scales, or you could infer that as preserving the coloration pattern of the mosasaur skin. Either way, this is from throat region of the mosasaur, and this is one of the finger bones of a mosasaur with this marking, scale marks on it. This seems kind of interesting structure to me because the throat of a mosasaur no doubt would have stretched a lot because they were eating a big animal items when they were alive. And so it would be interesting to wonder why this looks different from some of the dorsal part of the neck scales shown elsewhere, because the structure seems different. I mean, maybe this is distorted, but it doesn't look as regular as, for instance, what the scales shown here in A. It looks like it's almost you can stretch these rows of scales, but maybe that's only myself imagining the things. But who knows? We didn't discuss that in the paper. Moving along, other features on the body. This is the torso part. This is the left front flipper of the mosasaur. And right away, most of the bones are there. And here you see very short rib, and this is long rib. That's not because this rib has missed the distal part of the rib, but rather that's because this rib is indeed 50% in length from the anteriorly adjacent one. And we are going to look at some non-bone structure. And one is, well, two of them. Here we see this dark stained area, patches, this here and there. And what they are showing, and according to our chemical analysis, they have iron, carbon, and some other material in these red patchy parts. And what we think is that here it's representing the heart or liver of the mosasaur. And then this is here, this little corner close to the rib cage is what we interpreted as a kidney. And why kidney is so far forward in the mosasaur body, I'll explain in the next slide. And then here in this part, we see this kind of M-shaped structure with lots of small fish bones. And I think it was Mike who tells me that there are at least 4 different types of fish vertebrae preserved in this part. Now, what we interpreted this structure as was not a stomach contents, but rather gut contents. In fact, we think that this was an internal structure or even a cast of the colon. So that stomach was much further anteriorly, and then the colon was here. And that interpretation, having colon so far forward in this part of in the body of mosasaur, also ties into our interpretation of this kidney being so far forward in the body of the mosasaur. If you look at today's monitor lizard, which is also a anguimorphan lizard, here's the rib cage. Now the rib goes all the way through instead of stopping here, but the ribs were truncated so that we can see the internal structure. Here's the liver, here's the heart. So they're located quite anteriorly in the body of the monitor lizard. But when you look at the kidney, a colon, they are quite farther posteriorly, and this is one of its leg. legs. So really, those two features are seen towards the very end of the torso of a monitor lizard. Now, however, if you look at cetaceans, this is a dolphin, what happens is that compared to other mammals like us or cats or dogs, they have internal organs really pushed forward, including the kidney. Kidney is situated right posterior to the diaphragm, where in humans it would be somewhere around here. And same holds true for all the other organs, including digestive system. So having the colon a little bit anterior in this body section of the mosasaur, to us seems no surprise, given this streamlined body shape of a mosasaur approaching that of dolphins or other whales. And then we go into the rather interesting part of this specimen now, because this is the end of the specimen, and what we see here is the tail in a perfect or 98% perfect articulation, and yet having this conspicuous downturned curvature. It's bent. Now, before I talk more about the bend, I will just draw your attention once again to the skin structure. Here at the ventral side of the tail, the scales are vertically elongate. They are neither rhomboidal nor hexagonal nor diamond-shaped, as you can see in the other parts of the animal. So that's interesting, because we had very little knowledge about how the scales of mosasaur body might have differed from head to the tip of the tail. But that's interesting to see that it is fact that they were different from one part of the body to another. So the tail. All the conventional reconstruction of the mosasaur skeleton looked like them. Either if it's a different groups of mosasaurs, they reconstructed the tail as if it's a very stretched, long, sea snake-like structure. And if you look at more primitive group of mosasauroids, bigger group containing mosasaurs, what you see, this is a fine specimen of an Aigialosaur, I mentioned sometimes, where you have as many as 65 tail vertebrae, but with no bending. And so that is interesting. Well, that is at least similar to what the lizards used to look like on land. No lizards today on land would have a bent tail, and neither did this early version of a mosasaur. But if you start looking at other specimens of mosasaurs, either on exhibit, often on exhibit because they look nice, being a fully articulated skeleton, then you start seeing these specimens. This is a Tyrrell from Kansas, and you can see the neural spines are more or less evenly spaced. So, but at the same time, this tail column goes downwards. And whereas if you look at a specimen with a straight tail as a reconstruction, what you find here is that this strange wavy structure, so that you can have the straight tail, and also where this upward bend happens, you see the jamming of these poking neural spine structures, which to me suggests that it was a rather artificial rendering of the mosasaur tail, and not really truthfully showing what the tail used to look like in life. And I think this specimen shows it very well. This is the very same kind of mosasaur that we are talking about so the specimen we are reporting as Platecarpus. So you can see this discrepancy not from the riddle structure but from artifactual reconstruction or preservation. And to support those arguments, we also did some numerical measurements. And A shows so basically we measured the top part and bottom part of single vertebra, individual vertebra, and around the bend. So anterior to the bend, the length between the top and the bottom of each vertebra is nearly 1. So they are nearly the same. And that's the same, that holds true for the vertebrae that followed the bend. You can see it's close to 1. So the top part and lower part of the vertebra are more or less similar. Now, however, at the bend, what happens is that the top part is, I don't know if I can use the word significant, but they are larger, longer than the bottom part, so that it would really form a very, very gentle wedge shape. And the cumulative effect of having many vertebrae shaped like that is, of course, turning down of the column of the tail. And here is another experiment that we did, is that those three lines, the red line indicates the Mosasaur's tail, and then green this tail, and then blue one is this one. So what it's showing is that above this line, each vertebra is longer than tall. Now below this, it's taller than long. And you see very similar pattern between these two, and excluding this one, which has a very tall and short vertebra towards the base of the tail, and then it constantly or consistently increases its length. Whereas in Mosasaurs and this dog shark, it starts as a tall vertebra, becomes elongate. Otherwise, it starts as a long vertebra and then becomes shorter, but then taller back again. So longer back again. So that's the similar pattern you see in the organisms having this kind of shape where you have the bent morphology. So I think we are quite confident in having the Mosasaur reconstruction shaped like this. Well, we are certainly confident with the bend of the tail. And we further went into reconstructing this crescent-shaped tail fin that we think is a reasonable structure, soft tissue structure to reconstruct. The reason for that is if you look at modern day, for example, hammerhead shark, you see this crescent-shaped tail, but not all this tail is supported by bones. In fact, if you flip this over, you can make a very similar downturned tail, that of mosasaurs. And what you see is that this lobe, which is a ventral lobe of a shark, has no skeletal support, even cartilaginous support. In fact, sharks are cartilaginous. But what happens is that if you look at the orientation of these spines, where it has a vertical orientation to the long axis of the vertebral column, you find this very elaborate fin structure, soft fin structure. And then once you hit that point, this spine starts lying backwards again, and then there's no more fin lobe structure associated with that. So really, I think, and that's what we try to show, is that here on this finely preserved specimen of a mosasaur, the neural spine orientation is consistent all the way up to this bending area, where this neural spine becomes straight upward, and then becomes a little bit tilted forward and goes back to the straight upward, and then goes once again back to the posteriorly oriented posterior orientation. And so that where you have that straight orientation of the neural spine in relation to the axis of the vertebrae, we find it's reasonable to reconstruct the fleshy lobe fin, just to make this mosasaur look more like a fish rather than a eel. Eel is fish, but sea snakes, for example. So that's the process what we went stepped, and this is the reconstruction that was released when this paper was published last August. And this person, Stephanie, was also instrumental in providing us with those fine photographs, photographic images. But she certainly is a great artist as well. Now, I had a fortune of working with another artist that I introduced today in the early part, Professor Oda, in Japan, to make a more paleoecological reconstruction, including this tailed mosasaur. And I think I have that here, yes. So I didn't show you the tail end of this mosasaur in the first slide, but this is what he made. And some things that you can also observe here is not only the crescent-shaped tail, but also the fact that the mosasaur had very well-developed flippers, and Professor Oda also went into great details of reconstructing each scale on this part of the mosasaur, so that he can show the difference in the shape and size of the scales between the head region of the mosasaur and the body region of the mosasaur. And the coloration is also whale-like. Very dark dorsally and light ventrally. And you can also see this mosasaur really doesn't show the teeth on the upper jaw. Why? Because that's what we see in today's lizards or snakes. When you are chewing on something, you don't necessarily show those nicely pointy teeth as you can see when they are skeletonized. It's important to consider those factors when you are reconstructing life restoration. And I think this illustration illustrates that very well. Is that the end of the story? No. There's a bit more. Remember macroevolution and tempo and mode in evolution, which I wanted to talk at the end of this today's slide. So that's our Platecarpus there. And this is a geologic time scale, Triassic, Jurassic, Cretaceous. That's Mesozoic, and then going into Paleogene, the first part of the Cenozoic. The so-called fish shape with a deep and streamlined body and a crescent-shaped tail fin, not to mention the flippers instead of the limbs, seems to have evolved in different lineages of secondarily aquatic reptiles and mammals in the first 10 million years of the evolutionary history of those groups, and independently in different time periods. Ichthyosaurs did that during the Triassic. The marine crocodile Metriorhynchidae did that during the Jurassic. And then in the Cretaceous, mosasaurs came in, just caught up before their extinction, mass extinction. And then once the reptiles are gone, the oceans are peaceful and calm for some time, but then soon was reinvaded by a mammal group called cetaceans or whales. And they too evolved this streamlined, fluke-tailed body form that is like a fish in about 10 million years into their evolution after they became separate from their land-dwelling ancestors. So this means that these unrelated groups of vertebrates shared a similar tempo, evolutionary tempo, which is about 10 million years in this case, and mode, which in this case is acquisition of fish shape of evolution. And especially, we are often familiar with this mode of evolution. In other words, certain characters that you see in common in different groups of animals. But it is not that common to be able to deduce the tempo, the timing of acquisition of those interesting structures in big geological time scale in the very poorly, incompletely preserved fossil record. So that was something that we are really intrigued about when, as an idea that we could produce based on this latest addition of secondary aquatic tetrapod to this group of marine animals, tetrapods, because mosasaurs just singularly stood up as a great exception, not having this fish-shaped tail and having a very long, elongated, straight tail, but no, they are not exception. And with that, I'd like to thank you for my co-authors, Johan Lindgren, Michael Caldwell, and Luis Chiappe. As well, I thank the Royal Tyrrell Museum for the stimulating working environment. And finally, I certainly, well, not last, but second, finally, I thank my wife for continuous support and understanding of my work daily basis. And then finally, I really appreciate your attention today, and hopefully it wasn't too drowsy, and you're not thinking about lunch yet, just not yet. And then maybe have some questions you may want to ask me after the talk, and I would love to entertain them. So thank you for your attention.